So you see me if I stand up here? Okay.
Yeah, I should open this to make.
Okay, good morning, everybody, online and in person here in the room. It is a really great pleasure and honor for me to introduce today's guest speaker, Felipe Andrade, who I've known for quite a long time, and we've done some beautiful collaborations together. I hold Felipe in extremely high regard when it comes to rheumatoid arthritis research. So Felipe is an associate professor at the Division of Rheumatology at Johns Hopkins. He got his MD from the National Autonomous University of Mexico in 1990. He then did a residency and a fellowship in rheumatology, both also in Mexico, and then came to the United States in 2000 to work on a PhD thesis in immunology at Johns Hopkins. And he stayed on there as an instructor, became an assistant professor in 2005, and then was promoted to associate professor in 2014. And I do believe, he hasn't revealed anything, but I do believe he's up for promotion to professor. Maybe it's going to be announced soon. I'm looking forward to that. Felipe has 80 publications. He has a really long list of what I think are seminal observations in understanding rheumatoid arthritis and the role of citrullination and the PAD enzymes, and many of the things that we know really come from his research, and as well as his collaborators at Johns Hopkins, like Anthony Rose and Erica Darra. They've discovered really important things that my lab has been trying to follow and expand on in some cases. Felipe is very well funded. He has 2 R01s and at least 3 R21s at the moment. And I know there's multiple grants pending, and he's looking to expand his funding even further. And he's an unusual associate professor in that he does go into the lab from time to time, and he's still capable of doing experiments himself. His postdocs don't shoo him out when he wants to do a Western blot, because they know he makes the best Western blots out there. So with that, I'm inviting you, Felipe, to come up and talk about insights into the origin of ACPAs and perhaps the cause of rheumatoid arthritis. So welcome.
Thank you, Thomas. Good morning, everyone. Thank you for the nice introduction. You are always so nice to me. And you know, I've been working with autoantibodies for a long time. And since I have memory, how can I change? So since the time that I was a medical student, and there has always been this question of whether autoantibodies are pathogenic or not. And it's likely that some might be pathogenic, others may not be. But regardless of their pathogenicity, something that is remarkable with autoantibodies is that these are associated with unique disease phenotypes in autoimmune diseases. So you have anti-DNA in lupus, you have ACPAs in rheumatoid arthritis. In myositis, you can see associations of phenotypes with antibodies. So independently of whether the antibody is driving or not the disease, what this is telling us is that the antibodies are linked to a specific microenvironment, an antigenic microenvironment, that may tell us about the origin of these specific autoimmune diseases. So in particular, I'm going to talk today about ACPAs in rheumatoid arthritis. So ACPAs are a group of antibodies which are targeting a modification that is called citrullination. And they have several features which are interesting to know about the cause of rheumatoid arthritis. One is that these antibodies are antigen-driven. That means that the antibodies are highly mutated, meaning that they were exposed to the antigen to become antibodies with high affinity. And second, these antibodies are detected before disease initiation, before the clinical onset of the disease. That means that the antibodies may have the information and the memory of what happened just before the clinical onset. And if we understand that, we might be able to understand the cause of the rheumatoid arthritis. As I said, these antibodies target a specific modification that is called citrullination. Citrulline is a non-canonical amino acid that needs to be added to proteins after protein synthesis. And this process is mediated by a family of enzymes that they call the peptidine arginine deaminases, or the PAD enzymes, which are calcium dependent. So when the protein becomes citrullinated, then it becomes the target of these antibodies. So the PAD enzymes, there are 5 PAD enzymes, but there are only 4 which are functional. That is the 1, the 2, the 3, and the 4. And these enzymes are found in different tissues, and they target different proteins. Importantly, citrullination is a normal process, a process that is physiologic. Our body is full of proteins which are citrullinated. The brain, the lung, the skin is full of proteins which are citrulinated. However, something that even though there's a significant number of proteins which are citrulinated, there are a specificity about these proteins. It means that some of these proteins are only citrulinated in the brain, other in the skin, other in the lung. So despite that there's a huge number of proteins, truly, when you look at tissues, you can identify maybe 4, 5, 6, 10, 20 in a specific tissue. So there's a lot of specificity about how these proteins are generated. There's a huge list of proteins, more than 200, as I mentioned, but only in few cases we know what is the function of these proteins when they become citrullinated, and these are just some examples of how these proteins function. So when we knowing the antigen of these proteins, when we start working with rheumatoid arthritis, we wonder what is different in rheumatoid arthritis that make these proteins to be the target of antibodies that is different of just the whole citrullinome that is found in healthy controls. So the easiest approach that we took was to analyze the target tissue, that is the joint. And what we found is that the cellular content in synovial fluid from patients with rheumatoid arthritis is highly enriched with proteins which are citrulinated. This is a Western blot, and everything that you see in black are proteins of different molecular weights which are citrullinated and are detected with an antibody against citrullination, compared with osteoarthritis, in which you don't see much of that. So to this process, we call it cellular hypercitrullination. And using mass spectrometry, we and others have identified that the citrullinome in rheumatoid arthritis is quite extensive, and there are more than 200 proteins which are citrulinated. However, different of the normal citrullination, the citrullination that you see in the joints, here you have an accumulation of 200 proteins which are citrulinated at once, compared with tissues in which there are only few which are citrulinated. So the abundance, the magnitude of citrullination in the joint is quite high compared to what you see in peripheral tissues, in normal tissues. The process of citrullination in the joint is mainly mediated by neutrophils, which are the main cellular type that is found in synovial fluid, and is also known to be a cell type that expresses high amounts of the PAD enzymes, particularly PAD2 and PAD4. So when we look at, again, a very simple question using Western blot, that, as Thomas said, this is my power on the lab. We just ask if there's any specific mechanism in the neutrophil which may mimic or reproduce the hypercitullination that we see in the joints. And among different mechanisms of cell death in the neutrophil, including netosis, autophagy, apoptosis, we only found one that was able to reproduce the hypercitullination that is present in the joint, and is the induction of cell death by cytotoxic cells, particularly by perforin. We also found, and in fact in this assay we use killer cells to reproduce the hypercitullination, and we also found that complement is able to do this. Going further to look for other mechanisms that may drive hypercitullination, we found that different pathogens, for example, Aggregatibacter actinomyces incomitans, that is associated with periodontitis, and others like Staph aureus and pyogenes, also are able to induce hypercitullination. And particularly the mechanism involves pore-forming proteins. In fact, when you think about perforin, about complement, the mechanism is common for all these processes. And it's a quite simple mechanism. What happened is that the neutrophil is exposed to these pore-forming proteins. And since the calcium enzymes, since the PAD enzymes are calcium dependent, when you have these incredible flows of calcium inside the cell, the PAD enzymes become hyperactivated, and therefore that drives the process of hypercitrullination. So whether it is immune mediated by perforin or complement, or mediated by any kind of pathogen that has a specific pore-forming toxin, the mechanism is the same, that is by formation of this membranolytic damage that is mediated by pore-forming proteins. There are other pathogens that are also able to modify the normal citrullinome during infection. For example, CMV. This is quite interesting. The mechanism is by the induction of the PAD enzyme, so the virus infects cells and induce PAD expression. And in the case of CMV, CMV do that because when it activates citrullination, it citrullinate proteins that are involved in the antiviral activity, specifically IgGs. And in that way, the virus is able to survive during infection. There are not many broad analyzed citrullinomes in the literature, but among the few that are available, what you can see is that the proteins that are targeted in healthy controls in rheumatoid arthritis, in CMV, and by Aggregatibacter is quite different. And for example, in healthy individuals, citrullination target mainly proteins which have to do with structure of the cell. Aggregatibacter likes to target proteins that involve in apoptosis and so on. So when you compare the citrullinomes that are induced by different components, you can see that there are many proteins which are shared within the citrullinomes, and there are many others which are absent in healthy controls. So the view that the way that we see that is that there are these microbial-induced hypercitrulination, which is likely used as a mechanism to evade the immune system, in which you have this process of hypercitrulination, in which you have citrulination of proteins that are not normally citrullinated. And if you are genetically susceptible, that might be able to drive an immune response against this novel set of proteins that induce microbial agents. And this process is amplified when you have killing by killer cells, in which they mimic citrullinums that you can find during microbial infection. However, in this model of thinking about rheumatoid arthritis as a disease, or citrulination driven by some pathogens and by perforin and complement, the wonder is how the antibodies are made, and whether tolerance is truly lost against these autoantigens. So as a very simple model, when we see B-cell development, to explain the production of these ACPAs, there are many possibilities, and I'm going to mention only two of these possibilities. One is that we have a normal B-cell development, and because there are different conditions that are present during inflammation and during infection, you have the production of new proteins which are citrullinated. And as a consequence, a normal B-cell, a B-cell that can target anything, is going to evolve to target these new antigens, and then you will have a B-cell that is secreting or is producing these antibodies. As a second possibility, it is likely that there are defects in the central and peripheral tolerance, and therefore cells which are able or have the capacity to bind proteins which are citrullinated might be survived during development, and therefore these cells are going to expand when you expose the cells to these citrullinomes or abnormal citrullinomes that are generated during infection and during inflammation. So these are the two hypotheses that are potentially able to explain this process. However, and based on animal studies, not based on human studies, there are some caveats on this model. So some of the caveats are that at least in mice, citrullinated proteins are not really immunogenic. And there's a lot of controversy of whether citrullinated proteins can truly induce arthritis in mice. In fact, usually when it happens, the antibody response is not citrulline specific, but is targeting the protein in the native stage. And when people has look of why it can be happening, what has been found in mouse is that the repertoire of CD4 cells which target citrullinated proteins is really tolerized in the mice. And when you try also to make a transgenic mice expressing an ACPA, the cells that are expressing the antibody become anergic. So that means that at least in mice, there are mechanisms which are set up to avoid the target of proteins which are citrullinated, which kind of makes sense because citrulination, as I mentioned before, is a process that is normal. And as part of the development of B cells, you don't want to target autoantigens. Therefore, at least in mice, there's not evidence that this immune response against proteins which are citrullinated may exist because all the cells, either T cells or B cells, become tolerant to this. So to add more complexity to this model of rheumatoid arthritis, citrullination is not the only target of antibodies in these patients. And in fact, people has introduced the term of AMPA instead of ACPA as anti-modified protein antibodies. That means that the patient with rheumatoid arthritis not only target proteins which are citrullinated, but also target other modifications that can identify in these patients. And one of these modifications that is quite interesting is carbamylation. Carbamylation is a process in which lysines are modified to homocitrulline. The structure is almost identical, except that homocitrulline is one methylene group longer than citrulline. So the molecules are pretty much similar. So, and the truth is that when I was looking at when these antibodies against proteins which are carbamylated were initially described, because the similarity between citrulline and homocitrulline, I just considered it was just cross-reactivity. I mean, they're so similar that the most likely is that these antibodies are not important, and it's just the same antibody which is targeting the modification that is similar. However, mechanistically, citrullination and carbamylation have differences that might be important thinking about the mechanism of the disease. Citrullination is mediated by the enzyme. It's a process that is mediated by an enzyme, and it usually happens, it requires a reducing environment to efficient. Carbamylation is not. Carbamylation is a process that is non-enzymatic and is mediated during oxidative stress. More importantly, citrulination is a normal process. It's a process that is physiologic. It happens everywhere. It happens in development. In mice, it appears that the immune system is tolerant against proteins which are citrulinated, which is compared with carbamylation that is environmentally driven. So in this process of carbamylation, what happens is that isocyanic acid interacts with lysine and then modifies the residue to homocitrulline. And this happens during different conditions. It is induced during smoke. Smoking is a factor that is involved with the production of proteins which are carbamylated. And also during inflammation, isocyanic acid can be generated, and that induces carbamylation of proteins. So this process that is a chemical process has been associated with aging. This process accumulates through life, depending on how you are exposed to different factors that promote carbamylation, and has been associated, as I said, in the context of aging, physiological stress, inflammation. Proteins which are carbamylated, the function is altered. So carbamylation is truly a problem that happened and has been associated with different pathologies in humans. And more importantly, even though proteins which are citrullinated are not immunogenic, at least in the mouse model and also in other animals, carbamylated proteins are not tolerated, are highly immunogenic. And if you immunize mice with carbamylated proteins that are self, the mice develop antibodies against the proteins which are carbamylated. And in fact, there are models of arthritis that can be induced with proteins which are carbamylated. So that brings the question of, well, what has to do carbamylation with citrullinations? Is truly cross-reactivity or what? So to understand and try to get some insights about ACPAs and how these antibodies may originate, we decided to analyze monoclonal antibodies. Making monoclonals takes a long time, so the easiest way for us was to use monoclonals which were already available, in which the sequences were published. One of these monoclonals is called E4. This monoclonal is maybe one of the most best characterized monoclonals already in the literature. And the monoclonal was isolated from a patient with rheumatoid arthritis using citrullinated fibrinogen as the antigen. Initially, the antibody was called an anti-citrullinated fibrinogen antibody, but truly the antibody detects many antigens. It is a very high affinity using a peptide from enolase. And more importantly, the crystal structure of the E4 is known. So having all this information, we might be able to understand how carbamylated and citrullinated proteins may be interacting with ACPAs. So this project was developed by Marta Escarras and Marty. And again, based on Western blot, we had just a very simple question. So we used 293 cells, which we know that they don't have bad enzymes, and we transfect them to generate through activation with adenomycin, cell lyses that were hypercitrullinated. This is the control, and this is the activated cells. And you can see using an antibody against citrulination, how the assay works nicely to generate this hypercitrulination in the cells. And we use potassium cyanate to generate proteins which are carbamylated using exactly the same cell type. So you have carbamylated and citrullinated proteins. Then we run the lysates and we use the antibody E4 that we generate in the lab. And as has been previously described, the antibody is very nice. It's a very good ACPA that targets pretty much all proteins which are citrullinated, which is something that you see with the ACPAs, the antibodies. There are some which are more specific for some specific proteins which are citrullinated, but this one is kind of pan-citrullinated, pan-citrulination antibody. And the antibody has minimal recognition of proteins which are carbamylated. So means that the antibody is pretty much specific against proteins which are carbamylated. So then what we did is that we asked, well, based on the model that I was telling you, what will happen if we modify the antibody and we reverse the antibody back to the stage before the antibody had somatic mutations? That is called the germline form of the antibody. And I will refer to germline in the next slides. So what happens if we take out all the mutations of the antibody? And what we found, and is something that was already known, and for pretty much all ACPAs, is that when you take out all the mutations, the antibody can no longer detect proteins which are citrullinated. That means that the detection of proteins that are citrullinated are dependent on somatic hypermutation, and therefore these antibodies are antigen. However, guess what happened with the detection of proteins which are carbamylated? So surprisingly, the antibody now becomes fully specific for proteins which are carbamylated. So going back to the model that we said, well, maybe there's two possibilities. One is just that a normal B cell in a hypercitrullation environment goes, mutates, and generates ACPAs. Is not the case because this antibody is reactive from the beginning. And the second hypothesis that maybe there's some problem with the central and peripheral tolerance that allows the survival of an ACPA producing cell is neither demonstrated by this model. So what this model is showing that indeed all, at least for this ACPA, it originates from a germline encoded antibody, which was that the nature of the antibody was the targeting of proteins which are carbamylated. And when you think about that, it kind of makes sense. In the case of citrulination, as I mentioned, it's a process that is physiologic. Therefore, you have tolerance against citrulination. But for carbamylation, it's a process that is induced. Therefore, if during the cell development, you make antibodies against proteins which are carbamylated, there's no reason why you should delete them, because carbamylation doesn't exist within the development. Because, unless you have maybe the model is smoking or something, but carbamylation doesn't exist during development. Therefore, these cells these are just normal cells that are there and waiting for an environmental antigen to become activated. But still the question is about this thing that really citrulline and homocitrulline that is generated during carbamylation are pretty much identical. So how is that the immune system, how is that the antibodies are able to distinguish these two modifications? So for this, the best answer will come from making a crystal structure of the antibody with the antigen, but that takes too long. So that is why we decided to start collaborating with Jeff Gray and Michael Chongon, who are in the School of Engineering at Hopkins. And they do all these, they use these models to model structures and interaction with antigens. The challenge of this modeling is that even though it has done before the analysis of interaction between antibodies and antigens and specific antigens, the challenge here was that citrulline and homocitrulline are non-canonical, and the programs are not made to analyze non-canonical amino acids. So Michael, that is a graduate student in Jeff Gray's lab, worked on this and modified the programs to construct these non-canonical amino acids into the system to identify how these things interact. So they use 5 different structural programs to do this. And the one that was the best was Chai1. So Chai1 was the one that was able, and the advantage of this is that we have the crystal structure of the antibody. So the question was simple, is which one is the best system that is going to mimic what you see in the crystal structure? And the winner of this was Chai1, which, by the way, it is a preprint. These people that do these softwares for structural analysis, they just keep publishing preprints, and I don't know whether they cut the papers out, but it's still a preprint, and Chai1 showed that it was the best modeling system to understand the structure of the interaction of the antibody using a citrullinated peptide that we used before in the crystal structure that is based on collagen type 2. So this is a structure based. So once they were able to mimic the crystal structure using Chai1, then they modify the residues in the antibody to make the antibody a germline antibody. And this is the overlap between the wild type and the germline. The wild type is in green, and the germline is in blue. And when you superimpose both antibodies, you may not see much differences. But when you see the paratop pocket of the antibodies, that explains why one is specific for citrulline and the other is for homocitrulline. So the paratop pocket that in the germline antibody is wider, is bigger. So that allows the extra methylene group to fit perfectly into the pocket, something that that homocitrulline cannot do in a very tight pocket, that is the one that is binding citrulline. If you look at the surface charge of the antibodies, also the germline antibody is more negatively charged. There are differences in the antibodies, but for me, one of the most striking is the shape of the paratop where the residue is binding. By the way, the peptide the carbamylated peptide that we use is a peptide that indeed exists in the joint of patients with rheumatoid arthritis, and is a peptide and is also targeted by antibodies, a peptide from carbamylated alpha-1 antitrypsin. So for these models, for the citrulline, we use the peptide from collagen type 2, and which is also a target of antibodies, a very well characterized peptide. And for all the carbamylation studies, we choose the one from alpha-1 antitrypsin. We avoid to just change the citrulline to homocitrulline in the same peptide, because that's cheating. That is not fair, because that doesn't happen in nature. So when you look at the structure of the antibodies, and that is known from the crystal structure, the wild-type antibody has 3 critical interactions with citrulline that involves asparagine 105, serine 51, and tryptophan 48. All 3 residues are germline encoded. That means that during the process of somatic hypermutation, the antibody changes in the way that these germline encoded residues are in the correct position to interact with the citrulline. When you try to feed the carbamylated peptide into the wild-type antibody, it just doesn't fit. It doesn't fit. The model doesn't allow to fit, and likely because the paratope is so small that just the homocitrulline cannot go in. When you look at the structure of the germline, what is one of the most important changes is that tryptophan 48 in the heavy chain of the E4 can no longer interact with citrulline, because it is too far for the interaction. And that is a major critical component why this antibody lacks the specificity against proteins which are citrullinated. And not surprisingly, but similar to what we saw biochemically, now the germline antibody can fit perfectly the homocitrulline in the peptide. So it is known, and that is something that is not new, that tryptophan 48 in E4 is critical for the binding of the antibody to proteins which are citrullinated. So what we decided was to mutate this tryptophan to methionine, and indeed, when you mutate only one residue, this tryptophan to methionine, the antibody cannot longer bind proteins which are citrullinated, which is known, that has been published before, and that is because tryptophan 48 is critical for the interaction with citrulline. But guess what happened with the proteins which are carbamylated? A single mutation in tryptophan 48 makes the antibody to be non-specific to anything, to now to be a fully anti-CARP antibody. So what it looks like is that during the process of somatic hypermutation, the antibody shape allows these residues to be in the correct position to bind the citrulline. And tryptophan 48 somehow is in a position that determines whether the antibody is going to bind citrulline or it's going to bind homocitrulline, likely because this residue doesn't allow the homocitrulline to fit into the pocket, and therefore when you take it out, when you change it, now the homocitrulline fits perfectly. So about tryptophan 48, it's a quite interesting residue. It is in framework 2 in the antibody, and this is a residue that is conserving almost all variable genes, except for 2 that I don't remember now. But it's highly conserved, so this is germline encoded. And when you look at the sequences of a good number of ACPAs which are published, you can see that the tryptophan 48, that sometimes is in the position 47, sometimes in position 49, but is well conserved, is not mutated, is highly conserved, but not mutated in any of these antibodies, suggesting that this residue may also be critical and be a general residue important for pretty much all ACPAs. So we look at additionally 3 ACPAs which are also published. The sequence of the heavy and light chain are publicly available. These are called RA3, A22, and F12. And these ACPAs are antibodies that are well characterized as antibodies against proteins which are citrullinated, mainly using peptides. Something that we don't do in the lab. We don't use much peptides. We like to use proteins because it's cheaper, and also it gives you a broad view about the interaction of the antibodies with proteins. And it's interesting that these two antibodies, which are classified as ACPAs, when you use hypercitrullinated lysates or carbamylated lysates, they are pretty much carb specific. They don't target, or maybe they target few proteins which are citrullinated. And this antibody is specific for both citrullinated and carbamylated proteins. So now the view is expanding because you have antibodies against proteins which are citrullinated, carbamylated, and also you have these double reactive antibodies. And in the case of this specific antibody, again, when you revert the antibody to germline, again, the antibody becomes specific against proteins which are carbamylated. And when the antibody has a single mutation in I think this is tryptophan 47 or 49, what is the tryptophan? The antibody remains as an antibody against proteins which are carbamylated. In the case of these two that are targeting proteins which are carbamylated, when you revert them to germline, they remain as anti-carbamylated proteins, in which the reactivity is a little bit less because likely you are taking out mutations. But it's the idea is consistent that the origin of these antibodies, whether they are ACPA specific, whether these are double reactive or only reactive against proteins which are carbamylated, the origin of all these antibodies, the germline origin, is coming from a precursor that is targeting proteins which are carbamylated. Now, the issue with these antibodies, which are isolated antibodies, and you can argue that, is that potential mutations in the CDR3, we cannot change them because we don't know what has been mutated or not. So there's this group of antibodies that were published in 2018 by Tim Combe, and which are also available, the sequences, the light and heavy chain. And the advantage with this group of antibodies is that all of them are related and are coming from a single precursor. So each one of these antibodies are coming from a single precursor. And from this single precursor, we generate a germline. And with this, we were able, because even it's published, to reverse back to germline the CDR3. And when you do that, the finding is consistent, that the germline antibody for all these ACPAs are targeting only proteins which are carbamylated. And interestingly, with these ACPAs, when you see that they are all mutated and the different patterns of mutations, that when this germline antibody starts mutating, you can find all different combinations. Some of them lack reactivity to proteins which are carbamylated and gain reactivity to few proteins which are citrullinated. Some stay as more carb specific, double positive, and more citrulline specific. So what that is telling us is that from a single germline antibody, from a single one, you can generate the full spectrum of ACPAs that you can ever imagine. So it can be an antibody against enolase or vimentin or anything. So a single antibody, as it starts mutated, it can fully change what antigens are detecting. And I will say that you could explain the full ACPA production in a patient from a single germline that has mutated on different ways. So when you mutate one of these antibodies, that is this one, but only when you mutate the tryptophan to methionine, again, the antibody becomes only specific for proteins which are carbamylated. And this rule didn't apply for this one, in which we mutate also the tryptophan, I think 48 on this one, to methionine, and now the antibody didn't recognize anything. What it means is that although this feature of tryptophan 48 is common among many ACPAs, there are other residues in other antibodies that are likely important, that are playing a role also in the definition of what the antibody is detecting. For this specific antibody, it was quite interesting because as we increase the number of mutations, the antibody was shifting to become ACPA specific. So we thought, well, maybe there is an evolution of the antibody that as it accumulates mutation, it becomes more specific for protein which is citrulline. However, it was not the case for the second set of antibodies, which are also derived from a common germline. So these antibodies, all of them are related, are coming from the same antibody. When you make a germline again, and I mean, we can keep going with as many monoclonals as it will be cloned. So the precursor, again, is specific for proteins which are carbamylated. And in this case, the number of mutations was not associated with the specificity of the antibody. So some of these antibodies were more citrulline specific, which have less mutations, and some that have more mutations were double positive with carbamylation as the major target. And for one of these antibodies, that is the only one that we changed to methionine, the tryptophan, again, it becomes an antibody against proteins which are carbamylated. So what it looks like is that there is not continuum of saying the antibody is carb specific, becomes double positive, and then it becomes an ACPA. It is not the case. It appears that you have the precursor that is the anti-CARP precursor. And depending on the environment, the antibody can go to be a more specific for CARP proteins, or can be a double reactive, or can be an ACPA specific. But all of them are coming from a common precursor, which is targeting proteins which are carbamylated. There is a third modification that is acetylation that also has come as a modification that is importantly targeting patients with rheumatoid arthritis. And what acetylation has is that it is almost similar to homocitrulline, not to citrulline, with the difference that you have here, methyl group instead of an amino group that you have in the homocitrulline. So this modification has been proposed to be maybe even the target that initiates the whole immune response against proteins which are citrullinated. And when we look for all the antibodies against BSA, which is acetylated, only few of them showed activity with proteins which were acetylated. And it was just the E4GL and the E4 and the E4M and a little bit of this one of the RAM, but none of the others showed activity against acetylated proteins. So the conclusion is that based on this, acetylation is a modification that can be cross-reactive with some antibodies, but has nothing to do with the origin of these antibodies. So as a summary of this work, in the initial part, I showed that citrulination is dysregulated in rheumatoid arthritis and also is dysregulated during infection by microbial agents that are able to activate the paths, either by having pore-forming proteins or like CMV, which induces the expression of the path enzymes. ACPAs are very heterogeneous, and you have antibodies which are specific against citrulline, others that are specific against proteins which are carbamylated. You have these double reactive antibodies. However, all of them, independently of what they are targeting, when they are reverted to their germline form, all of them are antibodies against proteins which are carbamylated. And except for one antibody, the structural analysis suggests that the tryptophan 48 is a critical residue that is determining whether the antibody is going to be an ACPA or going to be an anti-CARP. And the data indicate that these antibodies, that ACPAs or anti-CARP or double reactive or whatever you want to call them, or against acetylated proteins, all of them are coming from a germline precursor which has a natural reactivity against proteins which are carbamylated. So just I want to finalize with this. All people show models, so I wanted to show this and how we view this. I think I believe that even though I don't work with mice and I'm not very positive about the work with mice, in rheumatoid arthritis, I believe that truly in the human system, the human model, B cells that have the capacity to target these proteins which are citrullinate are likely to be tolerized in the environment. So I don't think that they exist. In fact, when you look at the germline antibodies, they don't target proteins which are citrulinated. So we don't have precursors against proteins which are citrulinated. However, we have these precursors that are targeting proteins which are carbamylated, which, by the way, they are normal cells. So what you have here is not a loss of tolerance. It's just a cell that exists because the modification that is targeting is absent during development. So if this individual that has these cells is exposed and has the genetic susceptibility and is exposed to a microenvironment that is enriched of proteins which are carbamylated and citrulinated, the individual is going to end making these antibodies. So I will say that if the antibodies are the cause of the disease, this may explain how rheumatoid arthritis is originated from this set of antibodies, which are germline encoded. And the interesting part, and I think that Thomas is going to like that, it's not a lack of tolerance. It's just a normal response that you have from a precursor and an environment that drives these antibodies. So as I mentioned, this work was mainly done by Marta Escarzas and Marty, and we have this incredible collaboration with Jeff Gray and Michael Chongon, and I have a bunch of collaborators that have been involved in different things and different projects, and more importantly, truly this work could have been done with the help of the NIH. My lab is fully funded by the NIH. And I truly appreciate that. I'm from Mexico, and you have no idea how incredible it is to have an agency like the NIH. It's a blessing, truly. So thank you so much. And if you have any questions, I'm very happy.
What do T cells do? What do T cells see? Do they make the difference between
maybe, I don't know. You know, the T cells, different to the B cells, the T cell repertoire is limited because they cannot mutate. So for the T cell, I have always seen the T cell as a not very specific thing, despite that it is specific. And pretty much the T cells, if they have something about the T cell that I don't think that has been fully addressed is what the TCR is seeing. Is the TCR seeing the citrulline and the homocitrulline, or is seeing the back of the peptide? Because in principle, the idea is that the HLA is the one that sees the residue. So the TCR should not be able to have direct access to the modified residue. So then I think that T cells are helping this process to happen. But the T cell may not realize whether it's citrulline or homocitrulline. It might be just activating the system that thinks that is abnormal and may not be identifying the difference. And the second option is that you have T cells which are specific for each one. So either one works well, but and it works to help the B cells which are already existent in the host. I think that is the important part, and it's like what we're talking about, the lupus and CAR T cells. I mean, that shows that the B cells are the ones that are the major drivers of the disease. I think that in RA is similar. Is that unless you have these precursors, the T cells will do something. Otherwise, they do nothing. Yes, Dr. Elcon. Yes, kid. Yeah, no, there's no evidence. I think that the way that these antibodies have been managed is pretty much through associations. They people look at large cohorts of patients and they check the percentage of these antibodies, those antibodies, and they try to correlate with something. They are not pretty much different. I can say that I look at anti-carbamylated protein antibodies a long time ago in a cohort that we have, and 100% of the anti-carb positive patients were also ACPA positive. So my conclusion was that it's just correlativity. I don't think that there's any evidence that one might be more pathogenic than the other. For sure, what I can say is that the antibody hasn't been fully, when you see this, they have not been seen fully characterized. Because when I'm talking about germline encoded native antibodies against proteins which are carbamylated, in the patient or in the humans, you should expect that they are IgM, and nobody has looked at IgM antibodies against this protein. So then when you add these things of whether the IgM, IgG, or all that are more pathogenic or not, I don't know. My point of view is that the ACPAs might be more pathogenic, but I have no evidence for it. Yes. Yes. Well, I mean, I don't know whether I fully understand your question, but you see them. The problem with these systems is that when the antibodies are detected and we do it in the clinical setting to do associations, you have a plate that has proteins which are carbamylated and a plate of proteins which are citrullinated, and you test the serum in parallel, and you call them carbamylated and you call them citrullinated, but you don't know how much of them are overlapping. And as I show, there are many of these antibodies which are double reactive. So at the end, I don't think that, I don't want to think about pathogenesis. In fact, there are some evidence in mice that I don't consider that might be important, fully important for the human, saying that ACPAs are protective against rheumatoid arthritis in an animal model. So I don't want to think much about the pathogenesis, but I want to think more about the development of the disease and what these antibodies are telling us. And these antibodies, all this set of antibodies that are targeting these modifications, truly are just a combination of the same antibody in which the detection of one or another modification is dependent on how the antibody mutates. So once you have the precursor, you have two mutations in one, you have three in the other, and it becomes anti-carb, ACPA, or double positive. So all of them is just a continuum of this, of a single antibody that is mutated. So how ACPAs are involved in the disease is a question that is still kind of open. But if ACPAs are the cause of the disease by any mechanism, their origin for sure is from this precursor that is against proteins which are carbamylated, that, as I said before, it makes full sense. These cells, there's no reason why you should delete them. In fact, maybe if we take a sample from the people that is here in this room, and we look for anti-CAR P antibodies IgM, we might be able to identify many of us which are positive. And if you are a smoker, and if you are HLA-DR4, and you have periodontitis, that will push you to have rheumatoid arthritis. Otherwise, these antibodies are normally there in circulation as any other natural antibody that we have. But the point is that they are the source of the ACPAs. Yes. That is a very good question. In fact, and it's something that we wonder, not for carbamylation, but for citrullination, because I mean, citrullination is enzymatic, and in many enzymatic process, you have the reverse. Citrullination until today is considered that is not reversible, and how would be the name? So spetilaginase deaminase will be an antideaminase, I don't know, an enzyme that can reverse the process. It hasn't been discovered. And when you think about it, citrullination in that way as an enzymatic process might be easier to reverse. For carbamylation, nobody has looked at whether it is possible. I will say maybe, if you find it. But nobody has tried to reverse these modifications, which will be very good. I mean, independently of rheumatoid arthritis, carbamylation is involved on many things, in diabetes, kidney damage, aging, so inflammation, they accumulate over time. So if you can put them back, would be nice, back to normal.
So Felipe, there are some questions, 3 questions online here. The first one is from Susan Fink, who asks, does the model predict that germline anti-carbamylated protein antibodies would also be present in healthy controls?
Yes. Yeah. So I mentioned something of this. Yes, the answer is yes. Yeah. And specifically IgM likely. And if the healthy individual has been exposed to some environment like smoking, the chances that will be IgG will be higher. But yes, I will predict that these antibodies must be present in the general population. Yes. Very good question. Yeah.
So then we have 2 questions from Rayan Najjar. The first one is, citrullination is physiologic, so we expect tolerance against physiologically citrulinated peptides. But what if disease-specific citrullination develops in sites other than the physiologic ones? There would be no tolerance against such peptides, right?
Absolutely. And in fact, when we look at the model of hypercitrullination, that is what we thought, that by targeting, during hypercitrullination, there are 2 things. One, the magnitude of the proteins which are citrulinated is higher. And also, the enzyme starts citrulinating sites that shouldn't be citrulinated normally. So these are not neoantigens. But still, you have the question of whether a normal antibody is going to turn on to target these neoepitopes, or whether an existing antibody that has some reactivity is going to be the one that is going to drive the process. So the evidence that we have is that it is an existing germline encoded natural antibody against CAR-P, the one that is targeting these neoepitopes. So it's not, the model doesn't support a break of tolerance. It's just the activation of an existing B cell with neoepitopes that are produced by the environmental triggers, inflammation, and all that. Yeah.
Okay. So the last question here is kind of similar to what we heard earlier. So if ACPAs arise from anti-carbamylated antibodies, do you expect that anti-carbamylated antibodies should be more prevalent than ACPAs in RA patients?
No, because they switch. I mean, as I mentioned before, we have frequencies of anti-CAR-P using carbamylated proteins, and the frequency of ACPAs using citrullinated proteins. But we don't know specifically which ones are cross-reactive, which ones are specific. And even if we knew that, this is a continuum. This is a mix of antibodies coming from a single precursor, which is going to have variation in each one of the patients, depending on whether the patient maybe smoked more, whether the patient has more periodontitis. But this is just a continuum of antibodies, a mix of antibodies coming from the single precursor, that the difference is just the mutations, as you saw in the slides. The patterns of the antibodies change totally, coming from the same precursor. It's just the mutations what determines the specificity.
So we have one more question. Yeah. You used a citrullinated collagen peptide. Are they just a prototype or more common in RA?
Well, in the structure, we use the citrullinated collagen peptide because it was the one that was used in the crystal structure that was published. So we were looking for something that when we do the artificial intelligence model, we're looking for something that matched that. It was just because of that, because it was the one that was published. However, I want to specify that in all assays that we did for antibody binding, we use full-length proteins, which are citrullinated in cells. You saw the whole black thing in the Western. We don't know which proteins are these. The only thing that we know is that they're citrullinated. And we don't care much about the proteins because they are detected similarly. So, but yeah, in the model is because the peptide was in the original crystal structure. Yes. Sorry, say again.